So, DNA is found in all things from whales, like this whale's eyeball, to this diagram that I'm going to show you, and all the things that in it all had DNA at one point. All right, so for my model, I'm going to have my phosphates are going to equal an almond. That almond actually does have DNA in it right now. My deoxyribose sugar or pentose sugar will be a walnut. That also has DNA in it. And then my nitrogenous bases are going to be represented by Cadbury mini eggs. They do not have any DNA in them right now, but they came from plants. So there are 4 different nitrogenous bases. There are thymines, which are going to be represented by a pink Cadbury egg. There are adenines, which will be represented by blue. There are cytosines, which will be yellow. And then there are guanines, which will be white. All right. Now, a nucleotide is made up of all the different parts. So we'll put our deoxyribose sugar, our phosphate, and our nitrogenous base. Okay, so that would be our nucleotide. To build a DNA molecule, you have to covalently bond your nucleotides together. So what would happen is you would have your nucleotide, one nucleotide, and then it's going to covalently bond with another nucleotide. So here's one, here's another one. And what will happen is this phosphate is going to bond with that deoxyribose of that nucleotide, forming a sugar phosphate backbone. Sugar, phosphate, sugar, phosphate. All right, we'll do another one. Covalent bond between that almond and walnut, which is that phosphate and deoxyribose. Then our last one here. Move the chair up so you can see the full model. Now we have to use that as a template to make the other side of the DNA molecule. So I put the phosphate, the sugar, and then the nitrogenous base. So if this is a thymine, which is going to be a pink, all right, it's going to always match up with an adenine. So we get an adenine nitrogenous base. Oops. I'm going to put it right there. Now, what forms in between them is going to be a hydrogen bond, which will be represented by that highlighter. All right, another nucleotide. So if that's a thymine, that needs to have an adenine attached to it. So I have this nucleotide. Oh gosh, nitrogenous bases all over the place. Good enough. All right, so this will bond right here. Another hydrogen bond forms between them. All right, this phosphate bonds to that sugar. And this phosphate is already attached to that nucleotide. So then we would grab another. So this phosphate would bond to this sugar right here. That guanine will attach to this cytosine using a hydrogen bond. Oh, that nucleotide is not done. Now it is, because it has 3 parts, nitrogenous base, deoxyribose phosphate. Notice that these are going upside down too. They go in a different manner. Then we have to build our last nucleotide. That phosphate attaches to that deoxyribose, and then that cytosine will bond to that guanine using a hydrogen bond. All right. So those should represent hydrogen bonds that are forming between them using our food. All right. And now we have our DNA molecule. All right. So in this molecule, there are 8 nucleotides. All right. So there's 1, 2, 3, 4, 5, 6, 7, 8. In between each nucleotide is a hydrogen bond bonding the 2 nitrogenous bases. Between each of the nucleotides on one side, there's a covalent bond from the phosphate to the sugar of the next one. So all the nuts on here are forming your sugar phosphate backbone. All the Cadbury mini eggs are going to be your nitrogenous bases. And that is your DNA molecule.